Hi, in this video I'm going to provide a worked example to create a suitable file naming convention for biological data. And this is based on my own scientific research and microscopy data that was published in the following paper. I'll also use the study design as it is written in the abstract to support me in the design of a suitable file naming convention. Now, I'm teasing some good file name examples here. And deliberately, I don't want you or need you to understand the file names yet, but I want you to sort of hold on to the current confused feeling that you may have. And I'm doing so just to help reinforce the importance of documentation, which we're going to talk about at the end of the video. So we can create a naming convention in 7 simple steps, and these are defined in the file naming convention worksheet, which you can download in a link somewhere below. Credit is given to Kristen Briney from the Caltech library, who originally created the worksheet and which I've modified for our microscopy users. But first, if there exists already naming conventions in your lab or scientific discipline, then please use these. Existing naming conventions, they may already be well understood by your colleagues, so there's no reason for you to reinvent the wheel. But watching this video may help you to understand how that existing convention came about. So in the video, you can see the 7 steps listed, and I'll work through these systematically for my previous publication and my previous microscopy data. At the top of the screen, I've written out the study design as it was written in the abstract of the paper. And you'll see how this is useful to identify my specimen's most important attributes. But first, in step 1, it asks, what group of files will this naming convention cover? Often, a specimen may be imaged using different microscopes, so it is useful to know whether we use a single naming convention for images from different microscopy systems, and whether to include microscope information in the file name. If you're doing quantitative image analysis, often new images are going to be created from the raw image data at different processing steps, and therefore we need to decide whether the convention is recycled for each version of our image files. In my study, we used only a single microscope for imaging our specimen, and that is microcomputed tomography or micro-CT. These images are, however, processed, and thus the naming convention is going to apply to both raw and the processed image data. And I'll talk about tracking differences between my image files at a later step. In step 2, it asks, what attributes are most important to these image files, and to pick between 3 and 5. And this is where I'll now refer to the study design, which states that 14 weeks old female C57 black 6 and bulb C mice were divided into 2 treatment groups per strain, ovariectomized and non-operated control, with 6 mice per group. The right tibia was scanned at baseline, so 14 weeks old, and then every 2 weeks thereafter until 24 weeks old using in vivo micro-CT. It was easy for me to highlight the specimen and the experimental attributes as they are listed within that short passage of text. Now, as they appeared in order, we saw the animal age, the animal sex, genetic strain, species, treatment, mouse number, and anatomical location. So that's the right tibia, which is the bone in the lower segment of the mouse hind limbs. So 7 attributes to describe my specimens. But next, we'll decide that some of these attributes are more important to our file name than others. Remember, the question asks us to only pick between 3 and 5, and this ensures that our file names do not become excessively long, which may not be handled well by some software or operating systems. So, the most relevant or the most important attributes will be those that change among our specimens, and therefore change among our image data. We know that the animal sex, the animal species, and the image location, so the anatomy, are the same among specimens, and therefore we can discard these. That leaves mouse age, strain, treatment, and mouse number, and we have the beginnings of our first naming convention. Now at the bottom of the screen, I've shown some examples for different mice, and it was as simple as taking a description of the attribute or the attribute value and entering it within the naming convention. Now, as I already mentioned, it's important to keep the file name succinct. But as you can see with a verbose description, such as writing out the whole words ovariectomy, control, weeks old, that the file name starts to become many characters long. So the verbose description also means that in a list of files, the attributes, and a good example here is the mouse number, it does not well align from one image file to another. So I'm not seeing that mouse 1, that text, is aligned directly above text for mouse 4. And so at a glance, it's not going to be necessarily easy, or maybe a better word would be comfortable, to navigate visually and find the right files, particularly in a list of many hundreds of images. And we'll correct for this in step 3. So step 3, it asks, can you abbreviate or encode your attributes? Either can you reduce your attribute values to a 2 or 3 letter abbreviation, or can I encode an attribute, taking, for example, sex and assigning males the value 00 and females to the value 01? And of course, the answer is yes. For age, remember, from the study design, it is in weeks. We can abbreviate as W and some 2-digit number. So for example, 14 weeks old is abbreviated as W14, 16 weeks old as W16, etc. And similar for mouse strain and treatment group. C57 black 6 is BL6, bulb C is BAC, ovariectomized is OVX, and the non-operated control group is CTL. Now just briefly, a comment on abbreviations and something to keep in mind when handling your own attribute values. For ovariectomy, or OVX, it is the standard abbreviation, so that which is most widely used and accepted in the preclinical animal research community. Using it, it just means that when my data is shared with other researchers, they can interpret OVX, and even if they don't understand the rest of the file name, at least have a hint about the specimens from which I've obtained my image data. So the key message is, please use standard abbreviations and the nomenclature that's appropriate to your own scientific domain. And so we can now enter the abbreviated attributes or codes in our examples, and these file names become a lot more succinct. And because we consistently used 2 or 3 characters for each attribute, the attributes, when listed within our file directory, are now going to be well aligned. From our previous example, mouse ID number 01 is going to be in our list of file names well aligned above mouse ID number 04. So in step 4, it asks, how will you order these attributes in the file name? In step 2, we listed attributes as they appeared in our study design. So that was first, the mouse age, the mouse strain, treatment group, and mouse number. But this order, it might not actually be optimal when we think about how we want to use that data set. So you need to keep in mind when you default sort your files that the files are arranged in order, first numerically, then alphabetically by the file name and character by character. It just means a file called 00 comes before 01, a file called 1A is before AA, and a file AA is before AB, and so on. So the questions you need to ask yourself are, what files or what information do you want to see first? And how do you want your image files to be grouped? So in using my default attribute order, it meant that images were grouped together by the mouse age. All 14 weeks old images appeared together, then all 16 weeks old images, and so on. But operationally, this wasn't how I intended to analyze the data set. I was only interested at any one time in analyzing the mice according to their mouse strain. That way I could answer the question, how did surgery affect measurements made on black 6 mice, and how did those measurements change over time? Similarly, how did surgery affect measurements made on bulb C mice, and how did those measurements change over time? So when I default sort, I want to see all files for the white mice, the BAC, before seeing any files for the black mice, the BL6. And I therefore decided that strain was my most important attribute. I then prioritized all OVX mice over my controls, so the CTL, and consequently, in importance order, my attributes became strain, treatment, mouse number, and mouse age. So accordingly, I amended the naming convention and the example file names to reflect the change. And it's just noteworthy that the white BAC mouse appears now before BL6, which was previously sorted per the mouse age. So we've now spent a bit of time with these example file names. And of course, you're going to be less familiar with these file names than I am. But I think we can wholeheartedly agree that they're not a great pleasure to read. If you're following along in the video, then you've watched me build these file names step by step. But if you've just joined at this specific time point, it's not going to be clear where one attribute ends and the next should begin. For example, is the white mouse strain indicated by BA, by BAC, or BACC? Is the black by BL, BL6, or BL60? Let's improve this in step 5. So, step 5, it asks, how will you separate each attribute in the file name? And this step, it's going to ensure that the file names are easy to read. That is, for both yourself and for the computer. Now, to simplify it, please use only hyphens and underscores to separate your attributes. Spaces and other special characters such as exclamation marks, question marks, ampersand, $, hash, etc. They just don't play nice with some operating systems and image analysis software. It means you might not be able to find a file or open a file as required. So to iterate, hyphen and underscores only. For my image data, strain, treatment, and mouse number were separated by hyphens. And it is from those 3 attributes I could create a unique mouse or sample ID. And I can refer to that ID when talking with my collaborators. Then, underscores were used to separate remaining attributes. I update my naming convention accordingly. And now, if we look at the examples, there is a lot less ambiguity in our attributes. And I hope that you find these a lot more pleasurable to read. So in step 6, it asks, is it important to track different versions of each file? And I alluded to this in step 1, where the raw data is changed and new images are created at different steps in the image processing pipeline. So, in a typical micro-CT workflow, the raw or projection images are reconstructed, and the reconstructed images are likely filtered and then binarized. Now, I would like to recycle my file naming convention for each processing step. And so to track changes in the image version, the text raw, so RAW, rec, REC, or bin are inserted immediately following the mouse age in the file name and separated by underscore. And I can update my convention. We then amend our examples as necessary for raw and binarized images respectively. Now, there's still one aspect of the naming convention that I've deliberately overlooked, but you may have noticed in my naming convention the ellipsis, or often what you may call the 3 dots. And I've used it here to note the omission of certain attributes in the convention. And those are attributes often beyond your control that are decided by the microscope at the time you generate your image files. For my images, for my naming convention, I know I can replace the ellipsis with 4-digit slice number within the micro-CTZ stack, as it would be written by the microscope software. So I'd ask, please talk with your microscopist, and they'll inform you about missing attributes as would be written by the software. So well done. We've made it to the 7th and possibly the most important step. Write your file naming convention down. Do you remember the somewhat maybe confused feeling you had when I showed some file name examples at the start of the video? That, I guarantee, will be the universal feeling shared by your project PI, colleagues, or collaborators when you share with them your image data. But of course, you can help your colleagues by simply describing the naming convention, and we'll discuss a strategy to document your naming convention in more detail in Module 3. But for now, in a PowerPoint or a Word document, write your naming convention down. Write down an example, or better yet, examples of your file name. And, if reasonable, all possible attribute values. So in my case, the naming convention was strain-treatment-mouse number _ age _ image version _ image number .file format. And the mouse strain accepts values of only BL6 and BAC. For treatment, only values of OVX and CTL, and so on. And I've listed, where reasonable, remaining attribute values here. And that's it. When your colleagues ask for these files, you could simply share the images alongside this document that describes the naming convention. But now it's your turn. Complete the worksheet either in the MyUni portal or download the document from a link below and create your file naming convention for your own microscopy data. And I'll see you in Module 2, where I answer the question, how do I organize my image data? Thank you.